Hello everyone, welcome to another session of online learning video classes. And today in this session, we are going to study and discuss about Chapter 2, Unit 1, Diversity in the Living World. Chapter 1 is completed, that is the living world. Keep that in your memory. And now we go for Chapter 2, which is Biological Classification. In this chapter, the learning objectives that I am expecting from all of you is at the end of the session, you are able to define biological classification, methanogens, halophiles, thermoacetophiles, gram positive, gram negative bacteria, plasmids, epizones, etc. You are able to understand classification of kingdom Monera. You are also able to appreciate the role of cyanobacteria, blue-green algae, and the help that it renders in the field of agriculture. It's a great help to farmers. And then reflect on the role of mycoplasmas. So with this learning objectives in our mind, let us start the session. As I said in chapter 1, we studied about systematics and taxonomy. You may recollect that systematics is the science that deals with diversity of organisms and their comparative and evolutionary relationships and grouping of organisms at every level of classification, right from species to kingdom. Remember, right from species to kingdom, we made a ladder. And this way of classification of science based on identification, nomenclature, binomial nomenclature particularly, and classifying the organisms into groups is called taxonomy. So with that background, we move ahead, and we are thankful to this gentleman called Linnaeus. He is the father of the binomial nomenclature, and hence how is that we have done this ladder of arrangement. Like we started with the species, many species joined together form genus, and many genera form family, many families join together to form an order, and many orders will form a class, and many classes will be under one phylum, and many phyla will be under one kingdom. Like we have got two broad kingdoms, plant kingdom and animal kingdom. And as we move from species to phylum, the common characters will narrow down. So broad characters, one or two will be there in phylum. Whereas in species, as we have well defined, it has maximum characters and they live in a particular habitat and are able to reproduce and produce similar species or similar offsprings. So it's something like when someone asks you an address, we say a room or apartment, then floor, then which building, which city, and the city that comes in which street, and finally country and continent in the similar way. So the classification is done based on similarities and dissimilarities and other characters. So the most accepted classification right now is five kingdom classification. Of course, the people started with two kingdom classification, like broadly plants and animals. Later on, three kingdom, four kingdom, and most accepted one now is five kingdom classification. So in this five kingdom classification, we have five kingdoms: kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae, and kingdom Animalia. And under kingdom Monera, we have got two sub branches. They are Archbacteria or Archbacteria and Eubacteria. So let us learn about them. Particularly in this session, we are going to talk about the kingdom Monera. So kingdom Monera has got two branches, as I said, Archbacteria and Eubacteria. Arch means very old, It's an ancient bacteria. All ancient bacterial species come under this. Eubacteria is latest one. Eu means clear. They have clear in their physiology and anatomy. Look at the examples of archbacteria and eubacteria. Please read every picture that is shown to you, every photo that is shown to you, and then you will be surprised that on the point of a pin, look at the bacteria. How many bacteria will be there just on the tip of a pin? So this is how they spread. And the largest known prokaryote, it's a marine bacterium. Okay, Margareta. Look at that. And then there are these heat-loving prokaryotes. So we call them as thermophiles. Okay, and then we have got halophiles, that is salt-loving. They love salt. Their habitat is salt. And archbacteria, some of the examples like purple sulfur bacteria. Look at them. And eubacteria, of course, these are very dangerous fellows. Pneumonia, of course, anthrax. These are very two dangerous bacteria. And of course, where there is negative side, there is positive side also. And cyanobacteria, a blue-green algae, is very, very good bacteria for us, particularly in the field of agriculture. And they are of great help to farmers. We will discuss that in later. Now, what is the difference between archbacteria and eubacteria? In archbacteria, cell wall consists of peptidoglycan, whereas in archbacteria, it is polysaccharides and protein. In eubacteria, it has no histone protein associated with the DNA. The histone protein is associated with DNA. In eubacteria, a small, simple RNA polymerase, whereas in arch it is more complex. So we will talk much about this in future classes. Now let's come back to the original one, that is under kingdom Monera there are two groups, two broad groups, but totally there are 5 groups. They are archbacteria, eubacteria, and mycoplasmas, actinomycetes, etc. So we concentrate on archbacteria. So archbacteria, group of most primitive prokaryotes. Prokaryotes, I said karyon means nucleus, pro means primitive. So these bacteria will have very primitive nucleus, hence prokaryon or prokaryotes. So these are of 3 types. The first one is methanogens, second one halophiles, third one thermoacetophiles. What are methanogens? As the name suggests, methanogens are the bacteria which undergo anaerobic respiration. These bacteria produce a gas called methane. So we say methanogens are methane producing bacteria. So these bacteria convert carbon dioxide of swampy areas into methane. So these bacteria convert the organic substance present in cow dung into methane by fermentation. As all of you know, fermentation, gobar gas fermentation. Example, methanococcus. They are also present in the rumen of the cattle. What is rumen? Second stomach present in the cattle, where it digests the cellulose by fermentation and convert it into methane. Example, ruminococcus. Next, we come to halophiles. Halophiles, as I said just now, means salt-loving. These are found in extreme salty areas such as the Great Salt Lake and Dead Sea. Halophiles are surrounded by purple membrane in which a pigment, bacteriophodopsin, is found. Due to this reason, membrane absorbs the bright light and directly forms ATP, adenosine triphosphate. It is the currency of energy. Therefore, they are non-photosynthetic. So basically, halophiles are salt-loving bacteria. And next, we come to the third group, thermoacidophiles. Thermo means heat. Acido means acid. Thermoacidophiles are type of archaebacteria that like and love heat and acid medium. So heat and acid loving protozoans. These archaebacteria are found at those places where temperature is approximately 80 degrees to 100 degrees and medium is acidic. These are aerobic bacteria which have the capacity to oxidize sulfur to H2SO4. So sulfur is getting converted into H2SO4 at high temperature and at high acidic medium. Some of these bacteria are able to reduce sulfur to H2S under anaerobic conditions. They are found in hot sulfur springs such as Yellowstone National Park. Now we come to the next major group, Eubacteria. So in Eubacteria, as the name suggests, the bacteria have got reasonably good body structure. Eu means clear bacteria. So the Eubacteria has got two types, bacteria and cyanobacteria. So we come to cyanobacteria. As I said just now, cyanobacteria is the blue-green algae. These are photosynthetic prokaryotes with chlorophyll A, carotenoids and phycobilins. What are these? Chlorophyll A, carotenoids and phycobilins. These are pigments. Okay, they may be unicellular, colonial, filamentous and marine. So that means they are almost omnipresent, and some of them are terrestrial too. The colonies are generally surrounded by gelatinous sheath. Several cyanobacteria have the ability of carrying out nitrogen fixation. That's why I said these are very, very helpful to farmers and they help in agriculture, carrying out nitrogen fixation. Normally, farmers use a lot of fertilizers and the fertilizers will have side effect. Whereas when we use the spores of these bacteria, cyanobacteria, they will not have any side effect and nitrogen fixation takes place naturally. Hence, they are very good in the field of agriculture. The filamentous forms possess special cells called heterosis. Look at that heterosis in the diagram. And what are heterosis? These are the special structures present in cyanobacteria that fix atmospheric nitrogen. So nostoc and anabina are the examples, and you can always recommend to farmers these two so that they can spray the spores of nostoc and anabina and help in agriculture. Now chemosynthetic autotrophic, that is, these are non-photosynthetic autotrophs. Look at that, normally autotrophs means photosynthesis, but non-photosynthetic. Why? They use chemical energy instead of light energy for food synthesis. Normally we use sunlight. All the plants use sunlight through photosynthesis and produce ATPs. Here chemical energy is obtained from oxidation of chemical compounds, particularly sulfur compounds. So photolysis, what is photolysis? Photolight, lysis, breaking of water with the help of photons, light. So hydrogen is received from other sources like inorganic sulfur compounds, H2S, or organic compounds, amino acids, fatty acids, etc. Example, sulfur producing bacteria. Now heterotrophic bacteria. These are most abundant in nature. Hetero means different. They depend on others. Many of them have significant impact on human affairs. They are helpful in making curds from milk, production of antibiotics, fixation of nitrogen in legume roots, etc. Some are pathogens causing damage to human beings, crops, farm animals and pets. Cholera disease is caused by different cholera like Vibrio cholerae. Now we come to mycoplasma. Mycoplasmas are the simplest and the smallest of all the free living prokaryotes. They can survive without oxygen. Please remember this. This is UPSC question. Organisms that survive without oxygen, mycoplasmas. Due to absence of cell wall, the organisms can change their shape and are pleomorphic. Again, technical word. Pleomorphic. Morphology. Study of external features. Pleo, different. So the morphology keeps on changing. Hence pleomorphic. The organisms are often called MLOs, capital M, capital L, capital O, mycoplasma-like organisms. And sometimes they are also called PPLOs, pleuropneumonia-like organisms. I repeat, pleuropneumonia-like organisms, PPLOs. They form characteristic colony with fried egg appearance. Hence, mycoplasmas are very, very dangerous. Now, what are the prokaryotic shapes? You have already studied in your junior classes. Round, spring-like, comma-shaped, all this. Hence, we are also having in all of them some factors called F factor, R factor that help in reproduction. So, we come to the end of this session with kingdom Monera. So to recapitulate, kingdom Monera has got 5 groups: Archaebacteria, Eubacteria, Actinomycetes, Mycoplasma, and Rickettsis. And under Archaebacteria, you got 3 subgroups: Methanogens, Halophiles, Thermoacetophiles. And under Eubacteria, bacteria as well as cyanobacteria. And in bacteria, based on the stain that it absorbs, gram positive, gram negative. So with that, we come to the end of this session. I'll see you in the next session. Thank you.